Good day, everyone. This is Group 4 from Jose Abad Santos High School, and this is our video recorded of our final defense entitled Spoiled Food Bacteria as Fertilizer to Plant Growth. Group 4 is composed of RONARIE P. ENTOMA, FRANCHESKA IREE VILLADORES, LYNET BILLEN, and JUELA AMDAIN.
For the reasons to conduct this study, first, to focus on the studies of using spoiled food bacteria as fertilizer to plant growth for sweet basil plants. Second, to know if there's sufficient improvement in the plants after bio-natural fertilizer is applied. Third, to see the beneficial growth development of the plants. And last but not the least, to know if there is a significant relationship between the growth of sweet basil plant when applying fertilizer and natural growth process.
For our statement of the problem number 1, the question is, what proof of improvement in the plants is shown after bio-natural fertilizer is applied? As we can see, we have here the ANOVA table. Step 1 for it is the acquiring the mean of the group after planting sweet basil plants and applied mold yeast and spoiled milk as a bio-natural fertilizer. As we can see, we have here the list of the growth developments of plants starting from week 1 up to week 4. There is now a changes, as we can see, from the without fertilizer and applying bio-natural fertilizer. For the week 1 of without fertilizer, the average height of the sweet basil plant is 1 inch. For the week 2, we have here 2.1 inch. For week 3, 3.6 inches, and for week 4 is 4.7 inches. And for its mean, it is 2.85. For the spoiled milk, for week 1 of the applying spoiled milk to sweet basil is 1.26 inches. For its week 2 is 2.4 inches. Week 3 is 3.9 inches. And week 4 is 5 inches, with the mean of 3.14. Also with the mold yeast. After observing the plants within 4 weeks, researchers take down notes the growth development of plants with the usage of bio-natural fertilizer, such as spoiled milk and mold yeast. By the data presented, it shows the graph growth development using non-fertilizer and bio-natural fertilizer to sweet basil plants. Moreover, mean of each group is needed to acquire to use in the next step of our data. So here now is our next step of our data. This is the step 2, data of sweet basil plants using non-fertilizers, spoiled milk, and mold yeast. We have here the total number, which is 11.4, the mean, which is 2.85, and the summation of the total, which is 8.47 in the column of without fertilizer. And for the spoiled milk is 12.56, its mean is 3.14 and 7.92, as well as the mold yeast. By gathering all the data of sweet basil plants using non-fertilizer, spoiled milk, and mold yeast, it indicates the mean of each group, the squared of the given of each group, and the total givens. Moreover, the sum of squares and summation of x squared are also part of our data. The K represents the number of group column of the group number of the column, which is 3. And then for N, which shows the number of given, which is 4. By this data, we can now proceed for our next step. Step 3 is the completion of ANOVA table of sweet basil plant with non-fertilizer, spoiled milk, and mold yeast. The computed value is 4.55. Critical value is 4.26. Based on the ANOVA table we present, the results of our sweet basil plant starting from week 1 up to week 4 within the average total of 21 days, the different growth changes of our plants between without fertilizer and using spoiled milk and mold yeast to sweet basil plant shows a significance level. Therefore, if the computed statistic value, which is 4.55, is bigger than the critical value, which is 4.26, which is shown in the table, we will reject the null hypothesis. Then we can conclude that there is sufficient evidence to support our alternative hypothesis. Based on our findings, the use of spoiled milk and mold yeast as a natural biofertilizer has a result to make our sweet basil plant grow taller than using the natural growth process.
For research question number 2, in what way does bio-natural fertilizer such as spoiled food bacteria help to benefit the growth development of plants? During 4 weeks, calculated 21 days of observation, we found that the plant's development had improved significantly with fertilizer. Each picture shows the development of sweet basil plants' growth over time using spoiled milk and mold yeast. Biofertilizers are helpful microorganisms that increase nutrient availability for plants. By stabilizing the soil and increasing plant growth, it, it increases levels of vitamins, auxins, and other promoting compounds required by the plants. Organic waste from spoiled food can be composted or utilized as biofertilizer. Bacteria in spoiled food waste help biodegrade and compose. Bread has a higher nitrogen level than most other foods. As we can see, the easiest kind of bread to compost is moldy bread. When it's past its expiration date, though it's not though it's the ideal choice for composting moldy bread is much better. The fact that it has mold on it indicates that it has already begun to decompose on its own. Adding moldy bread to your compost is nothing more than helping nature. Whatever variety of bread you have, if you're not going to eat it, it can be a tasty treat for the bacteria in your compost. Bread and spoiled milk both help in the growth of plants. Spoiled milk contains proteins, vitamin B and C, and carbohydrates that benefit plants and increase crop yields in general. For the soil, it is also good for the bacteria to feed on milk's fertilizer components. Now let's proceed for the statement of the problem number 3.
Statement of the problem, question 3. The growth comparison of plants while using spoiled food bacteria such as spoiled milk mold yeast as fertilizer and natural growth process of plants. This the data represent the growth changes of sweet basil plants using non-fertilizer each fourth day per week. It shows changes when it comes to the length of the plants. At 0.6 cm in the fourth day, 1.1 inch in the 8th day, 2.8 inch in the 12th day, 2.5 inch in the 16th day, 3.2 inch in the 20th day, 0.43 inch in the 24th day, and 4.7 inch in the 28th day. This data the growth development of plants by using different types of fertilizer such as spoiled milk and mold yeast. The pair of graphs show the growth comparison. So our first appearance, the non-fertilizer soil with sweet basil peaks in length for 4.7 inches within 4 weeks of development. Therefore, using of bio-natural fertilizers with basil as an 1 inch in the 4th day, 1.5 inch in the 8th day, 2.2 inch in the 12th day, 16th day 2 inch, 20th day 3.5 inch, 24th day 4.5 inch, and the average of height 5 inches in the 28th day. On the top of that, the usage of bio-natural fertilizers with basil has fast growth changes of plants every 4th day per week.
For the reference, this is the site that we gather to find more evidence to support our research paper. And that is how we end our video presentation for our final defense. Thank you.